Hello and welcome back. In one of my previous videos, I covered the prokaryotic cell in detail. However, many of the human pathogens can be unicellular or multicellular eukaryotes, and therefore, in this video we will study the eukaryotic cell. Only certain eukaryotes are traditionally studied by microbiologists, primarily the protozoa, fungi and helminths. All protozoa are unicellular. The algae and fungi can be either unicellular or multicellular. Helminths are truly multicellular organisms. The birth of eukaryotes on the earth is a mystery. Approximate entry of eukaryotes dates back to 2 billion years ago, while prokaryotes were born 3.5 billion years back. One of the theories explains the birth of eukaryotic cells by endosymbiosis. Large prokaryotes engulfed smaller prokaryotic cells. These smaller cells begin to live and reproduce inside the larger cell rather than being destroyed. As the smaller cells took up a permanent residence, they started performing specialized functions for the larger cell, from serving as nucleus to performing functions such as food synthesis and oxygen utilization. Many of these endosymbionts enhanced the cell's versatility and survival. Over time, the engulfed bacteria gave up their ability to live independently and transferred some of their genes to the host cell. It is now well known that mitochondria of eukaryotic cell is something like a tiny cell within a cell. Some bacteria that were engulfed developed symbiosis with the cell and developed to become mitochondria that is capable of independent division, contains a circular chromosome that has the bacterial DNA sequences and has ribosomes that are clearly prokaryotic. Mitochondria also have bacterial membranes and can be inhibited by drugs that affect only bacteria. Eukaryotes were so versatile that they soon spread out into available habitats and adopted greatly diverse styles of living. The first eukaryotes were single-celled and independent, but over time they began to aggregate, forming colonies. With further evolution, some of the cells within colonies become specialized or adapted to perform a particular function advantageous to the whole colony, such as locomotion, feeding or reproduction. The cells of a eukaryotic organisms are so varied that no one member can serve as a typical example. This image presents the generalized structure of typical algal, fungal and protozoan cell. The general cell organization of a eukaryotic cell is as follows. External components can be either appendages or glycocalyx. The appendages include flagella and cilia, and glycocalyx includes capsule and slime layer. The cell envelope components include cell wall and a cell membrane. The internal components are the main differentiating feature from the prokaryotic cells, and they include membrane-bound organelles inside the cytoplasm that includes nucleus, endoplasmic reticulum, Golgi body and mitochondria. In addition to these, eukaryotes have ribosomes and cytoskeletal components inside them. As with the prokaryotes, we begin with the external components and proceed inward towards the cell. So starting with the locomotor appendages in the eukaryotes, that are cilia and the flagella. This is an electron microscopic picture of a cilia on a eukaryotic cell. And this is an example of a flagellum helping a eukaryotic cell to swim in liquid. Cilia and flagella motility allows a microorganism to locate nutrients and to migrate towards positive stimuli such as sunlight. It also permits them to avoid harmful substances and stimuli. Locomotion by means of flagella or cilia is common in protozoa, algae, fungal and animal cells. Although they share the same name, eukaryotic flagella are much different from those of prokaryotes. Prokaryotic flagellum has a hook between the basal body and the filament. The hook can rotate clockwise or counterclockwise, thus moving the bacteria in different directions. Eukaryotic flagellum does not have any hook. It comes straight from the plasma membrane. It is 10 times thicker than the prokaryotic flagellum and has S-like motion. The bacterial flagella, on the other hand, have screw-like motion. Both flagellum and cilia have the same structure containing microtubules in regularly spaced 9+2 pattern in the entire extent and are responsible for the movement of flagellum. Cilia have back and forth beating motion like a whip. Cilia are very similar in overall architecture to flagella, but they are shorter and more numerous. Some cells have several thousands of cilia. They are found only on a single group of protozoa and certain animal cells. Such protozoa are among the fastest of all motile cells as seen in this video. Most eukaryotic cells have a glycocalyx, an outermost boundary that comes into direct contact with the environment. In this picture, you can see the endothelial cells of blood vessels having a glycocalyx layer that comes in contact with the blood components. This structure, which is sometimes called an extracellular matrix, is usually composed of polysaccharides and appears as a network of fibers, a slime layer or a capsule, much like the glycocalyx of prokaryotes. Because of its positioning, the glycocalyx contributes to protection, adherence of cells to surfaces, and reception of signals from other cells and from the environment. The nature of the layer beneath the glycocalyx varies among several eukaryotic groups. Fungi and most algae have a thick, rigid cell wall surrounding a cell membrane, whereas Protozoa and Helminth cells lack a cell wall and have only a cell membrane. The cell walls of fungi and algae are rigid and provide structural support and shape, but they are different in chemical composition from the prokaryotic cell walls. Fungal cell walls have a thick inner layer of polysaccharide fibers composed of chitin or cellulose and a thin outer layer of mixed glycans. The cell walls of algae are quite varied in chemical composition. Substances commonly found among various algal groups are cellulose, pectin, mannans, and minerals such as silicon dioxide and calcium carbonate. The cell membrane of eukaryotic cell is a typical bilayer of phospholipids in which protein molecules are embedded. In addition to phospholipids, eukaryotic membranes also contain sterols of various kinds. Their relative rigidity makes eukaryotic membrane more stable. This strengthening feature is extremely important in those cells that lack a cell wall. Cytoplasmic membranes of eukaryotes are functionally similar to those of prokaryotes, serving as selectively permeable barriers. Membranes have extremely sophisticated mechanisms for transporting nutrients in and waste and other products out of the cell. Now moving on to the main differentiating feature of the eukaryotic cells, that is the internal structures. Unlike prokaryotes, eukaryotic cell contains a number of individual membrane-bound organelles that are extensive enough to account for 60 to 80% of their volume. The control center or the nucleus is a compact sphere that is most prominent organelle of a eukaryotic cell. It is separated from the cell cytoplasm by an external boundary called the nuclear envelope. The envelope has an unique architecture. It is composed of two parallel membranes separated by a narrow space, and it is perforated with small regularly spaced openings or pores formed at sites where two membranes unite. The nuclear pores are passageways through which macromolecules migrate from the nucleus to the cytoplasm and vice versa. The nucleus contains a matrix called the nucleoplasm and a granular mass, the nucleolus, that stains more intensely than the immediate surroundings because of its RNA content. The nucleolus is the site for ribosomal RNA synthesis and a collection area for ribosomal subunits. The subunits are transported through the nuclear pores into the cytoplasm for final assembly into ribosomes. A prominent feature of the nucleoplasm is the stain preparation is a network of dark fibers known as chromatin that actually comprises the eukaryotic chromosomes. The chromosomes in the nucleus of most cells are not readily visible because they are long, linear DNA molecules bound in varying degrees to histone proteins, and they are far too fine to be resolved as distinct structures without extremely high magnification. During mitosis, however, when the duplicated chromosomes are separated equally into daughter cells, the chromosomes themselves become readily visible as discrete bodies. Endoplasmic reticulum is a passageway in the cell. It is a microscopic series of tunnels used in the transport and storage. Two kinds of endoplasmic reticulum are the rough and the smooth endoplasmic reticulum. Electron micrographs show that the rough endoplasmic reticulum originates from the outer membrane of the nuclear envelope and extends in a continuous network through the cytoplasm, even all the way out to the cell membrane. This architecture permits the spaces in the rough endoplasmic reticulum, called cisternae, to transport material from the nucleus to the cytoplasm and ultimately to the cell's exterior. The rough endoplasmic reticulum appears rough because of the large number of ribosomes partially attached to its membrane surface. Proteins synthesized on the ribosomes are shunted into the inside space or the lumen of the rough endoplasmic reticulum and held there for later packaging and transport. In contrast to the rough endoplasmic reticulum, the smooth endoplasmic reticulum is a closed tubular network without ribosomes that functions in nutrient processing and in synthesis and storage of non-protein macromolecules such as lipids. Golgi apparatus is the packaging machine of the cell. The Golgi apparatus, also called the Golgi complex or body, is the site in the cell in which proteins are modified and then sent to their final destinations. It is a discrete organelle consisting of a stack of several flattened disc-shaped sacs called cisternae. These sacs have outer limiting membranes and cavities like those of endoplasmic reticulum, but they do not form a continuous network. This organelle is always closely associated with the endoplasmic reticulum, both in its location and function. At a site where endoplasmic reticulum meets the Golgi apparatus, it buds of tiny membrane-bound packets of protein called transitional vesicles that are picked up by the forming face of the Golgi apparatus. Once in the complex itself, the proteins are then modified by the addition of polysaccharides and lipids. The final action of this apparatus is to pinch off finished condensing vesicles that will be conveyed to organelles such as lysosomes or transported outside the cell as secretory vesicles. Nucleus, endoplasmic reticulum and Golgi apparatus together form a nature's assembly line. As the keeper of the eukaryotic genetic code, the nucleus ultimately governs and regulates all cell activities. But because the nucleus remains fixed in a specific cellular site, it directs these activities through a structural and chemical network. This network includes ribosomes, which originate in the nucleus, and the rough endoplasmic reticulum, which is continuously connected with the nuclear envelope, as well as smooth endoplasmic reticulum and Golgi apparatus. Initially, a segment of the genetic code of the DNA containing the instructions for producing a protein is copied into RNA and passed out through the nuclear pore directly to the ribosomes on the endoplasmic reticulum. Here, specific proteins are synthesized from the RNA code and deposited in the space of the endoplasmic reticulum. After being transported to the Golgi apparatus, the protein products are chemically modified and packed into vesicles that can be used by the cell in a variety of ways. Some of the vesicles contain enzymes to digest food inside the cell. Other vesicles are secreted to digest material outside the cell, and yet others are important in the enlargement and repair of the cell wall and membrane. A lysozyme is one type of vesicle originating from the Golgi apparatus that contains a variety of enzymes. Lysosomes are involved in intracellular digestion of food particles and in protection against invading microorganisms. They also participate in digestion and removal of cell debris in damaged tissues. Other types of vesicles include vacuoles, which are membrane-bound sacs containing fluids or solid particles to be digested, excreted, or stored. They are formed in phagocytic cells in response to food and other substances that have been engulfed. The contents of the food vacuole are digested through the merger of the vacuole with the lysosome. This merged structure is called a phagosome. Now coming to mitochondria, which are the energy generators of the cell. Although the nucleus is the cell's control center, none of the cellular activities it commands could proceed without a constant supply of energy, the bulk of which is generated in most eukaryotes by mitochondria. When viewed with light microscopy, mitochondria appear as round or elongated particles scattered throughout the cytoplasm. The internal ultrastructure reveals that a single mitochondria consists of a smooth continuous outer membrane that forms the external contour and an inner folded membrane nested clearly inside the outer membrane. The folds on the inner membrane, called cristae, may be tubular like fingers or folded into the shelf-like bands. The cristae membranes hold the enzymes and electron carriers of aerobic respiration. This is an oxygen-using process that extracts chemical energy contained in the nutrient molecules and stores it in the form of high-energy molecules or ATP. The spaces around the cristae are filled with a chemical complex fluid called the matrix, which hold the ribosomes, DNA, and a pool of enzymes and other compounds involved in the metabolic cycle. Mitochondria are unique among organelles in that they divide independently of the cell, contain circular strands of DNA, and have prokaryotic sized 70S ribosomes. These findings have prompted some intriguing speculations on their evolutionary origins from prokaryotes. Ribosomes are the protein synthesizers. They are numerous tiny particles that give a dotted appearance to the cytoplasm. Ribosomes are distributed throughout the cell. Some are scattered freely in the cytoplasm and cytoskeleton. Others are intimately associated with the rough endoplasmic reticulum as previously described. Still others appear inside the mitochondria. Multiple ribosomes are often found arranged in short chains called polyribosomes or polysomes. The basic structure of eukaryotic ribosomes is similar to that of prokaryotic ribosome. Both are composed of large and small subunits of ribonucleoproteins. By contrast, however, the eukaryotic ribosomes is the larger 80S variety, that is a combination of 60S and 40S subunits. As in prokaryotes, eukaryotic ribosomes are the staging areas for protein synthesis. The cytoskeleton is a support network of the cell. This image shows microtubules of a cell fixated in a gel. The cytoplasm of a eukaryotic cell is crisscrossed by a flexible framework of molecules called the cytoskeleton. The three main types of cytoskeletal elements are the actin filaments, intermediate filaments, and microtubules. This framework appears to have several functions, such as anchoring the organelles, moving RNA and vesicles, and permitting shape changes and movement in some cells. The cell is a crowded place, so one of the functions of the cytoskeleton is to act as a highway along which cellular components can be moved to the proper part of the cell. This movement is driven by motor proteins like kinesin, which walk along microtubules, pulling their cargo along with them. The combination of different cytoskeletal components and different motor proteins allows the cell to control exactly where signal molecules or other proteins are delivered within the cell so that they can have the proper effect. Actin filaments are long, thin protein strands shown here in red. They are found throughout the cell but are most highly concentrated just inside the cell membrane. Actin filaments are responsible for cellular movements such as contraction, crawling, pinching during cell division, and formation of cellular extensions. This picture shows a mouse fibroblast cell. This cell has been stimulated with a growth factor that activates a signaling pathway leading to lamellopodia for protrusion. The protrusion of the cell membrane at the front of the cell is one of the first steps in cell motility. The actin cytoskeleton, that is a critical component of the skeleton of the cell that drives cell migration, is labeled in green, and lamellopodin, an actin regulatory protein, is labeled in red. Intermediate filaments are rope-like structures that are about 10 nanometer in diameter. Their name comes from their intermediate size between actin filaments and microtubules. Their main role is in structural reinforcement of the cell and of the organelles. For example, they support the structure of the nuclear envelope. So that was all about the eukaryotic cell in this video. Hope you liked the video. Please like, subscribe and share. Keep watching and supporting my channel. Thank you.